Good morning, everyone. It's really a great pleasure to be here with you today, and I would like to thank the organizers for inviting me for this important meeting. The topic of my talk is bispecific T-cell engagers and emerging therapy in the management of non-Hodgkin lymphoma. So during the coming 20 minutes, I will try to cover main pivotal trials that resulted in approval of bispecific T-cell engagers in DLBCL, diffuse large B-cell lymphoma, follicular lymphoma, and touch base on mantle cell lymphoma, starting with short background on non-Hodgkin lymphoma as unmet medical need, going through the historic development and structural differences of bispecific T-cell engagers. So as you all know, lymphoma is a heterogeneous disease, and diffuse large B-cell lymphoma is the most common aggressive form of non-Hodgkin lymphoma, represented approximately 30 to 40% of non-Hodgkin lymphoma patients. Follicular lymphoma is the most common indolent, representing approximately 20% of non-Hodgkin lymphoma. Mantle cell lymphoma represents approximately 6%. It's less common than CLL, but it is a focus of our talk today because the field of bispecific T-cell engagers is more advanced in mantle cell lymphoma than CLL due to duration. They are two stable specific B-cell antigens that represent a great potential therapeutic target. The story and the development of monoclonal antibodies went through many attempts that started in 1975. And with the development advancement also of novel technologies, the possibility and the success to have the first monoclonal antibody that targets CD20 that was proved to be effective and also safe, the rituximab, the first monoclonal antibody against CD20, was approved in 1997. To remind you with the mechanism of action, rituximab works through direct activation of complement system, so complement-dependent cytotoxicity. It works also by activating natural killer cells, antibody-dependent cytotoxicity, and also through the circulating or free fragments of complements recognition by macrophage, together with the FC portion recognition of rituximab by macrophages activating phagocytosis, another form of antibody-dependent cytotoxicity. The last mechanism of action is by the cross-linking of rituximab on the surface of tumor cells, resulting in its direct apoptosis. So for the first three mechanisms of actions, remember that the FC portion is important in the recognition of effector immune system. With advancement in the management of non-Hodgkin lymphoma, we still have unmet medical need in the treatment of non-Hodgkin lymphoma, because you can see on the left side of the slide, 20 to 25% of diffuse large B-cell lymphoma are still challenging and possess a very dismal event-free survival and overall survival. These patients are either primary refractory or refractory to second-line therapy, or relapsed after autologous stem cell transplant. 20% of follicular lymphoma are also challenging, especially if they progress within the first 2 years of treatment. The same for mantle cell lymphoma, and you can see on the left side that overall survival in patients who relapse after BTK inhibitors, in this case ibrutinib, you can see that the median overall survival is approximately 8 months. Regardless salvage therapy. Indeed, with the development of CAR-T therapy, there is improvement in outcomes of these patients in mantle cell and diffuse large B-cell lymphoma, but the problem is CAR-T cell therapy is associated with significant adverse events in terms of neurological, cytokine release syndrome, neutropenias, and recurrent infections. Not only that, CAR-T cells is not available for every patient, and it requires lymphodepletion, and therefore not every center can provide patients with CAR-T therapy. The success of immune-based therapy that was unfortunately coupled with toxicity and also unavailability to every patient opened, however, the door for research regarding other immune-based therapies, and hence came bispecific T-cell engagers. If you look at the middle of this slide, you can see that bispecific antibodies are over-the-shelf man-made antibodies. They have two single-chain variable fragments, recognize one epitope in the tumor cell surface, and by the other epitope, our immune effector T cell, CD3 positive. The Fc portion of the antibody is silenced, so it's not active, and therefore antibody-dependent cytotoxicity and complement activation is not part of the mechanism of action of T cell engagers. If you look at the left side, you can see that T cell engagers act by migrating and attracting effector T cells within the tumor microenvironment, causing direct tumor lysis. On the far right side, you can see that in normal immune response, the major histocompatibility complex is required for T cells to function. T cell engagers bypass the major histocompatibility complex requirements and engage the T cells directly with tumors. Not only within the tumor microenvironment, T cell engagers also attract and migrate CD3 from the circulation within the tumor microenvironment, resulting in more tumor cell killing. So what are the current clinical data that supported the approval of bispecifics in non-Hodgkin lymphoma? Before we start, I would like you to have a look on these bispecific T cell engagers that are in advanced stage of development: mosunetuzumab, glofitamab, epcoritamab, ordonexamab, and plamotamab. You can see that they are mainly IgG1 structure, except the ordo is IG4. Most of these bispecific T cell engagers are based on the CD20 epitope of rituximab, ofatumumab, or obinutuzumab. The administration in most of them is intravenous, except for EPCO. It is from the beginning subcutaneous, and currently mosunetuzumab is transferring from intravenous to subcutaneous routes. The adoption of fractionation of the target dose, what we call step-up dosing, allowed the development of T cell engagers because it reduced the cytokine release syndrome and immune-related toxicity at the CNS level. Most of these T cell engagers have 1-to-1 ratio, one antigen epitope related to the tumor cell, the CD20, and one to the effector T cell. However, glofitamab was developed to have 2 recognition sites for the tumor cells versus 1 for the effector T cell. And this was the basis to improve efficacy, but it was also a concern of increased cytotoxicity and associated cytokine release syndrome. And for that reason, we will see now that the mitigation process with glofitamab is much more intense. The first in development that was approved in relapsed refractory follicular lymphoma is mosunetuzumab. It was first in class. I will provide here more details about the study design because it's the basis of all T cell engagers. So these patients are usually relapsed after at least 2 lines of therapy. They have to have been exposed to CD20 and 1 alkylating agent. The first cycle includes the administration in fractionation. So cycle 1, day 1, then day 8, and by day 15, the target dose is reached. Then at cycle 2, the target dose is given. And from cycle 3 to 8, from 60 milligram, we move to 30 milligram as the maintenance dose. Patients who are in CR at cycle 7, they are allowed to stop treatment, while patients in PR or stable disease, they should continue to 17 cycles. So the development is finite therapy, fixed duration, and not till progression. And this is very important. The primary endpoint was CR that was compared to historic control of 14%. So the patients' population in this study was really of high risk population. The median age was 60. They were mainly males, but it was mainly advanced disease. The majority received at least 3 lines of therapy, including CAR-T in 3%, also autologous stem cell transplantation more than 20%, but more than 50% of this patient population were double refractory to CD20 and the alkylating agent. They were also progressing within 2 years of treatment So very challenging and very refractory population. The response rates were impressive, 80% overall response rate for all patients, CR 60%. And with this CR rate that was highly significant compared to the historic control, the drug was approved in follicular lymphoma. What's also interesting, if you look at the right side, the response rate in the double refractory population and also on patients who progressed within 24 months is impressive and is still high, reaching 70% or more for double refractory and 85% for early relapses. In terms of safety, mosunetuzumab is considered one of the well-tolerated bispecific T-cell engager. Let's start on the right side. You can see that the CRS rates is 44.4%. And you will see now with glofitamab, it's much more higher, exceeding 60%. The majority are grade 1, 2, and 3. Very few are grade 4, and none was grade 5. The median time of onset was few hours from the first injection and within 24 days from the target dose. The median duration for the complication to resolve was 3 days, so a very short duration. And few patients had to receive corticosteroids, and only approximately 8% received tocilizumab. In terms of neurological toxicity, it was mainly grade 1, 2, and 4% of patients. And as common in all bispecific T-cell engagers, neutropenia occurred in 1/3 of patients, and few were serious in 20%. The FDA approval of mosunetuzumab was also taken into account into consideration the results from CAR-T therapy, because you can see on the left side the median duration of responses in responders and also in patients in complete remission is very high, approximately 2 years in responders, and not reached with more than 1 year of follow-up in patients with CR. This is really considered favorable compared to CAR-T therapy because this is a single agent, well tolerated, and also available to all patients. Subgroup analysis within the pivotal trial was done for elderly patient population, and you see on the left side of this slide that patients older than 65 years obtained impressive overall response rate of 87% and more than 70% to complete remissions. In terms of safety, this patient population also were tolerating well, even had lower CRS rates, and few of them required treatment, and none was grade 4. So for the sake of the time, I will also show you the study of mosunetuzumab in frontline elderly patient population above 80. So this is a very frail patient population who usually in the clinical practice are not able to receive any treatment, if ever many are CHOP with poor outcomes. The study included also patients above 60 with comorbidities. The first report of 54 patients was impressive, with overall response rate approximating 60% and 43% CR. Patients achieving complete remission was really durable over a year. And neutropenia within 15%, not a single patient had neurological toxicity. CRS was only in 1/4 of patients that was resolved quickly. So to remind you, the mini R-CHOP is not applicable to most patients, only and approximately 60% are unable to receive treatment in this patient population. What about glofitamab? Glofitamab, as I said earlier, that it is built with a 2-to-1 molecular configuration with double linking to the CD20 and one linking to the CD3. And therefore, in the design of trials for glofitamab, it required more mitigation with the inclusion of obinutuzumab, the anti-CD20, 1 week before treatment and sometimes the same day of treatment if patients are high risk. The treatment is given intravenous and it's a finite therapy, so fixed duration for 12 cycles. Primary endpoint was CR.
So again, the patient population was even much more high risk compared to the population in mosunetuzumab, and you can see that the majority of patients received more than 3 lines of therapy, CAR-T in more than 30% of patients, and many of them failed in a short time CAR-T therapy. CR rates were more than 39%, with overall response rate exceeding 51%. You can see also that this was significantly higher compared to the historic control of 20%. The median time to CR is very short, within a month and a half. And the CR rates were consistent even in CAR-T exposed patients, and was higher in patients with relapsed disease compared to refractory population. In terms of toxicity, on the left side you can see the majority of toxicity is related to cytokine release syndrome followed by cytopenias. On the right side you can see that the majority of toxicities indeed were grade 1 and 2, but the rate is very high, exceeding 60%. Median time to CR was approximately 2 weeks. Many patients, more than 1/4 of patients require corticosteroids, and more than 1/3 of patients require tocilizumab. Neurological toxicity, despite less than 4% had grade 3, but indeed is still a concern. Overall, it was completely resolved in this patient population, and none of patients discontinued or died due to immune-related toxicity. The CR was consistent in patients receiving CAR-T, and you can see that the probability of progression-free survival and overall survival is considered favorable in this patient population, and 78% of patients achieving CR remain in complete remission. The approval of glofitamab occurred June this year. So it was also tested in mantle cell lymphoma with impressive response rate. As you can see here, 83% overall response and 73% complete remission, even in patients who are exposed to BTK inhibitors on the right side of the slide. Moving to our last part with EPCO. EPCO was tested in relapsed refractory diffuse large B-cell lymphoma, and it was the first subcutaneous T-cell engager to be examined. EPCO is given till progression, so it's not finite therapy, and it produced more than 60% response rates, and more than approximately 40% were in complete remission. Median progression-free survival is not reached with more than a year of follow-up, and the FDA granted accelerated approval for EPCO for relapsed and refractory diffuse large B-cell lymphoma in May this year, and the EMA is considering a positive feedback for the submission. Reminder, compared to the other first two regimens, it is treatment till progression. In conclusion, the bispecific T engagers have emerged as very effective anti-lymphoma therapy. Mitigation plans to cytokine release syndromes and immunological associated neurological toxicity were effective. This year, the FDA granted accelerated approval for the 3 drugs in diffuse large B-cell lymphoma, follicular lymphoma. The field now is moving towards combination therapy and also treatment in the frontline setting. And with that, I would like to thank everyone for the attention.
Thank you, Dr. Safa, for the elegant presentation and for the updated presentation. Actually, I want to ask you about the first-line bispecific antibody in combination, as we still have unmet need for increasing the CR rate, especially in the IBI high-risk group.
This is a very good question, and actually it's of great interest to researchers, and there is a very wide range of frontline studies, some of them with chemotherapy, some of them with BTK inhibitors, like in the case of CLL and mantle cell lymphoma, and some of them also with lenalidomide and tafasitamab in diffuse large B-cell lymphoma, and also in diffuse large B-cell lymphoma in combination with R-CHOP. So the field is going for frontline therapy. Some of them already started right now, and some are going to be starting at least early next year.
The expected CR rate in this group of patients?
Okay, there's two things here. The first thing is, you know that, for example, R-CHOP or R-CHOP polatuzumab is really providing high rate of CRs. So even if with this kind of therapy, you can obtain the same rate, more or less, of CRs, but with more durable responses, more prolongated progression-free survival, this can be an achievement to compare, of course, to other immune-based therapies. So no less than 80%, and most of the studies, 90% to complete response rates is the target.
Dr. Safa, thanks a lot. Highly appreciated your presence with us in this conference. My question related to, we have unmet need to complete the question of Dr. Soraya. And we have in the market CAR-T, Polivy, etc., which is a more and more appealing treatment for diffuse large B-cell. In your opinion, regarding the best position of each?
This is a very tricky question. Okay, so the vision of CAR-T and T-cell engagers in that context is based on the type of patient and also on the center capacity and resources to do the CAR-T. So, for example, we know that for CAR-T, the patient should go to lymphodepletion treatment before reaching CAR-T because they have to be responding, or at least with some kind of cytoreduction. The collection of T cells sometimes is compromised by previous lines of therapy. So the timing for patients is very critical, and the availability of resources in the center to manage toxicities. So we believe that T-cell engagers would be a good alternative for, number one, a very aggressive patient who does not have time to go through all the process for preparation for CAR-T, or for wide distribution on over-the-shelf available treatment across all centers in countries that are also not having this possibility. So I think it's the type of patient, the center resources, as well as I believe the necessity to finite therapy in these patients and the strategy to finite therapy is going to make the competition very high with CAR-T therapy.
Okay, thank you, Professor Safa, for this very elegant presentation. And now we come to the second speaker, Professor Ayman Saad. Dr. Ayman Saad is a professor of the School of Medicine of Al Faisal University. He is the section head of acute leukemia in King Faisal Specialist Hospital and Research Center in Riyadh, Saudi Arabia. He is a former professor of internal medicine, Ohio State University, and is the founder chair of NCCN Committee on Hematological Stem Cell Transplantation. Professor Ayman will speak about the development of CAR-T cell therapy. Dr. Ayman, please.
Thank you so much. I appreciate the invitation. I'm very happy to be with you all today. I'm trying to share my slides. So how I'm going to share a screen? I'm still disabled from sharing the screen. Maybe I can send it in the chat and someone opens it. So, here you go. Now I can share the screen. Okay, so do you all see this now?
Okay, you can see it.
Okay, so let me make sure it's progressing. So I'm going to tell you, I enjoyed actually the talk of Dr. Safa Ramadan about learning about the bispecific. This was very interesting. But I'll see if I can make a case for the CAR-T business here. So I'm going to tell you how CAR-T cell was developed by telling you initially just a couple of stories, and then we'll talk about how to develop a CAR-T program. So as we all know, the evolution of cancer therapy has evolved over decades, not necessarily for one modality to replace another, but basically trying to find complementary therapy that is more effective and less toxic. And I believe immunotherapy was created for the same purpose. So we'll start. What I'm gonna call the Boston story. In 1968, a 63-year-old man admitted for cholecystectomy. The surgery went well, except for one extraordinary fact. The surgical resident noted that 12 years ago, the same patient was admitted in the same hospital with terminal stomach cancer, and he went to the OR, was found to have multiple liver metastases. And he was sent home to die in hospice. And even he didn't die, he came back to have cholecystectomy after 12 years. So he had what we call a spontaneous resolution of his cancer. And this was described here by Dr. Steven Rosenberg, who was actually the resident who described this story. So the phenomenon of spontaneous regression of cancer was actually observed in both preclinical and clinical setting. This nice experiment done in Cornell in early 1900 showed that when they infused the lymphoma tumor into a group of dogs, there was some dogs, actually maybe one dog in this experiment, who didn't develop the lymphoma. I'm going to call this the immune dog. They transferred the blood, just plain blood, to the other dogs, and surprisingly, they found one of the dogs actually had the tumor disappearing in it. Not only that, but when they attempted to reinfuse the lymphoma cells again, this dog remained immune against developing lymphoma. In the clinical setting since the late 50s, again, spontaneous regression of tumors was also observed in several cancers. So going back to Steven Rosenberg in NIH, who pioneered what we call adoptive immunotherapy or cell transfer therapy in cancer, he developed two kinds of immunotherapy. He started first by what he called lymphokine-activated killer cells. He isolated the lymphocytes from the patient in the animal model who was using the spleen, exposed this or activated them in vitro by IL-2, and infused this to the patient along with IL-2. And then he said, well, instead of just taking any plain lymphocytes from the circulation, he can go after the lymphocytes that infiltrates the tumor, assuming that they have a special predilection to attack the tumor cells. And he called this tumor infiltrating the lymphocytes, or TIL, and again dissected these lymphocytes, activate them in vitro with IL-2, and then infuse this back to the patient. And he had actually very nice responses. There's a patient with melanoma infused with lymphokine activated, and you can see some response. And another patient with melanoma also having lung metastasis that regressed, and here was after infusion of TIL therapy. And then people have taken this a little bit further, and they said, well, instead of just looking at plain lymphocytes randomly attacking cancer, maybe we can find a T-cell specific clone that attack cancer, and this was the idea of antigen-specific cytotoxic T cell. And we know that the T cell recognize cancer cell by recognizing the foreign antigen expressed on the HLA. It has to be HLA class 1 if it's a CD8, for example. So they isolated these reactive T cells, isolated the gene, and packaged this gene in a cell machinery and made a lot of T cells, infused the cells back to the patient. And this is what we call antigen-specific T-cell therapy. And again, it showed some response. This is a patient with bad plasmacytoma and had some regression with this T-cell receptor T-cell therapy. But here is a problem. The problem is, as I said, these T-cells only recognize antigen presented by HLA class 1. What if the cancer antigen is not expressed by the LHA molecule? And this, the solution for this problem was hypothesized again in Steven Rosenberg lab, when they thought they can genetically engineer a T-cell receptor that's a chimeric or synthetic that will recognize the cancer antigen independent from the HLA expression. So instead of the normal process here, without any intervention, you can just hypothesize that you can make a synthetic receptor that recognizes the cancer cell and kill it. So that's obviously a sort of cell therapy, immunotherapy, and gene therapy. But this was in the late 90s. And in the late 90s, there was a huge setback that happened to the gene therapy experiment. That's why I'm going to come to the next story. And briefly, that was a patient who was born in Arizona with an important error of metabolism that caused liver dysfunction, yet he survived to be an adult. And he volunteered to enroll in this study with gene therapy when they injected an adenovirus vector carrying the correct gene. And this was a phase 1 study in the University of Pennsylvania. JC developed severe immune response, multi-organ failure. Unfortunately, he died 4 days after the adenovirus injection, adenovirus vector injection. And this was obviously in the news, 1999. Unfortunately, after this, things had just gotten worse. In France, similar therapy has induced leukemia in little kids being treated by gene therapy. So the US and France regulatory authorities came together and decided to hold any gene therapy at that time. So this was in 2002. And of course, scientists continued to work in the vaccine. And here we'll come to the how CAR-T came to the scene. So going back to Steven Rosenberg's idea, manufacturing this synthetic receptor that can recognize the cancer cell. So this is the recognition domain here, and this is the signaling domain, CD3 here inside the T cell that can kill this cancer cell. Problem with this therapy at that time in the preclinical model is the limited efficacy. These cells worked for a little bit, but not sustainable. And it was not until Michel Sutherland in Memorial Sloan Kettering Cancer Center, he developed what we call now the second generation CAR-T. So Michel said, well, since the T cell function is heavily regulated by co-stimulatory signals and inhibitory signals, why don't we just have one of these co-stim signal and insert it in this chimeric receptor and see if it worked? And that's exactly what he did. He actually selected CD28, which is one of the co-stimulatory signals, and put this in tandem with the signaling domain, and he found robust and sustained, that's the most important thing, sustained efficacy of this CAR-T therapy. He shared this idea with the people in NIH, UPenn, and NIH actually treated the first patient who is what they call Axi-cell in 2009. They were using the same co-stimulatory domain that Michel was using, CD28. UPenn used a different domain for 1BB and developed a different CAR-T product called T-cell and infused the first patient just a few months after the NIH did. And that was the first FDA-approved CAR-T product. So, and UPenn published their data actually in the New England Journal of Medicine, just one case report. And this was a CLL, low-grade lymphoma, received this magic CAR-T, and you can see here later on, the patient went to complete CR. And this patient actually is still in CR until today, as far as I shared a few years ago. These nice CAR-T show up in the peripheral blood as atypical lymphocytes, and can also show in the CSF. Actually, as a matter of fact, they often show in the CSF. And then we'll go back to what I'm calling fun story, and this back to where the failure started. And that's again an Arizona native, but this time is Emily. And Emily was diagnosed with ALL when she was 5 years old. Had chemotherapy, achieved CR1 to that part, to that extent, that is great. But unfortunately, 17 months later, she relapsed. And she went to UPenn looking for a CAR-T therapy, because that was the time when CAR-T was starting to be developed. And there was no trial open at that time, unfortunately. So she went back to the oncologist in Arizona. She had chemotherapy again, and she failed to respond. Hospice was recommended. The parents didn't give up. They decided to go back to UPenn. And at that time, they met Stephen Grubb. And Stephen Grubb was just opening the phase 1 study of the CAR-T cell therapy. And Emily received 3 doses of this experimental CAR-T cell therapy. After the 3rd dose, she developed a high fever, respiratory failure, and she was in a shock. They had to intubate her, put her in the vent, 3 pressors, and the situation was just a mess. So remember, this is in 2012, and back to 1999, that's exactly 13 years. And you know what 13 means in the Western world. So 13 years ago, a patient also in the same hospital, that's UPenn, remember that was UPenn, died with this similar scenario. Gene therapy infused, patient had a catastrophic death, okay? So Steve Grubb at that time, he looked at the whole picture and he said, well, clinically that looks like what we call cytokine release syndrome, but we don't know how to prove this or even don't know even how to treat this. Luckily enough, the part of this protocol actually required a chicken cytokine analysis in these people, but the turnaround time was about 2 weeks because that's non-actionable lab workup. So Steve Grubb called the lab. He begged them to just run this quickly. He needs to know what's her cytokine profile. And they actually, luckily enough, 2 hours later, they called him and they told him her IL-6 was 1,000-fold higher than her baseline. So, okay, now we know for sure this is cytokine storm clinically and lab-wise. It was 3 p.m. And that's their usual time for multidisciplinary meeting in the Children's Hospital of Philadelphia, CHOP. And they were not clear what to do. We know this is IL-6, we know this is CRS. And she already had high-dose steroid. The only known therapy for cytokine storming at this time was no improvement. The only way that they could have gotten out of this is just to give an anti-T cell therapy, like ATG, high-dose cytoxin, anything that kills this CAR T, this crazy CAR T, and go back to square one. She will eventually die of her leukemia. And that probably this is the only thing that they would do. So Steve Grubb reached out to his colleague, Carl June, who's a pathologist and scientist who developed this CAR T in UPenn, and actually Carl June was out of town, of course. He was giving a talk in Seattle, so he called him and he explained to him the situation that Emily is basically dying with what looks like cytokine storming. And Carl June on the other side of the phone told him that his daughter has juvenile rheumatoid arthritis, and she actually received a monoclonal antibody called tocilizumab. And they both thought maybe they can try this to antagonize IL-6. Okay? And luckily enough, the CHOP hospital had actually tocilizumab stocked, and they decided to give it. At 8 p.m., which is 5 hours after being in shock and on the vent, they infused the tocilizumab. And Emily began to improve dramatically, and she was weaned off vasopressor. As a matter of fact, the nursing staff decided to throw her a third birthday party a few days later. And the story says that she regained her consciousness, opened her eyes while the nurses were singing together, "Happy birthday to you." But this was like a farewell birthday, assuming that's going to be her last birthday on the earth before she goes to heavens. She remained awake. 8 days later, she had a bone marrow biopsy that showed no leukemia. So Emily's still alive, and she celebrated her birthday until now. She turned 18 recently. But there are a lot of ifs here that, if you can think about, you think, oh my God, things could have gone wrong here easily. Imagine if the cytokine profile was not being tested in the study. Imagine if the result didn't come on time. Imagine if even Carl June's daughter was not in tocilizumab and being treated, and things could have been different. Had Emily died, the CAR T-cell therapy would have likely died with her, said Stephen Grubb and Carl June. As a matter of fact, both Grubb and June, they believe that Emily wouldn't survive that night. Carl June actually, he stated that he even started drafting a letter to the provost of UPenn, because, as I told you, this could have been a disaster. So why is this story important? So remember, Emily was treated in 2012, and that's 2 years after the initial infusion of CAR T in NIH or UPenn. And it took the NIH, it took the FDA 7 years to approve the first CAR T. This tells you how much vigilance they have, and they were not really sure if this is going to be an effective therapy. And the most important thing about the story is this was the first use of tocilizumab in CRS, which is now a standard of care to treat CRS regardless of the etiology. Right now we have 6 FDA-approved CAR T-cell therapy targeting CD19 and PCMA for different types of lymphoma and multiple myeloma. Well, the good thing about CAR T, maybe the selling point I'm going to call it, is the persistence of efficacy. It's one-time treatment, very expensive, but not everyone responds. You get 60 to 80% response. Of course, CR rate here with this study was very high, was very nicely 50%, but it was sustained in about 40%, assuming the hypothesis is this therapy may cure up to 40% of patients. And remember, there is no curative treatment that we've known at that time, except for autologous transplant curing maybe 30% and allo maybe curing 50%. And this is a Zuma study again, updated in 2019, showing again this longer follow-up here. You can see the plateauing of the survival curve. And again, a last update published in 2023 for the same Zuma 1 study, showing continued plateauing. Nice plateauing. I'm not calling it plateauing, but semi-plateauing in the cell-to-cell product. That's a multiple myeloma product here. But the only caveat with multiple myeloma is this is short follow-up. As a matter of fact, if this CAR T can show plateauing at 5-7 years in multiple myeloma, I believe this is going to be revolutionary, because we don't know anything that can cure myeloma right now except allogeneic transplant. And of course, autologous transplant, we know that all people progress on labs. So, how long do I have here? Let me see. But I'm going to quickly talk about the CAR T program and how to develop a CAR T program here. So, most important thing for CAR T program, in addition to the routine clinical service, apheresis, cell processing facility, which is not really different than what you would have for any cell therapy-based product, is having management guidelines. Because as Dr. Safar Ramadan thankfully stated in the previous presentation, this therapy can be very toxic. As a matter of fact, it can be life-threatening. Around 3% of people can die because of toxicity. And this doesn't happen with the bispecific, I have to admit. And this is basically the CRS, as we saw here, neurotoxicity, cytopenia can even be a disaster. So there must be very clear guidelines to how to handle this patient. The journey of the treatment itself is not sophisticated. You freeze the T cells, you manufacture them in a central facility, you treat, and that is the most important part when you follow up these people. So, but the main problem is not the process and not even the clinical management, because this can be all accomplished. The major problem is the cost. The commercial product now, the cost, as you see, like close to $500,000. And that is a check that you cut for the company, aside from the cost of the medical care. So is this sustainable? Well, in my personal opinion, that's not a universally approved opinion. I think it is for developed countries, people who develop CAR T. That's what I mean by developed here. But people who don't have a CAR T, non-developed CAR T, who are going to just paying a huge amount of money. So I don't think this is sustainable for people who don't have CAR T manufactured in companies locally. So how can we get access to the CAR T? Let's just logistically think about this. People can, if you have a center anywhere in the world, in non-US, non-Europe, they can enroll patients in international clinical trials and get the product for free. But this is not going to happen until this center becomes a center of excellence and show that they can successfully infuse CAR T with success. The other thing, local manufacturing. I'm going to tell you in a second. Medical center can actually manufacture the CAR T, and that's going to cost 1/100th of the cost. So instead of $400,000, this can be only $4,000 per product, or even less. The other thing that we can get fancy and develop local pharmaceutical company that produce a commercial product in the country. And India has done this. Immuno Institute company, spun-off company, pharmaceutical company, that came out of the India Institute of Immunology or something like that. They actually now have a commercially available CAR T product in India, and they sell it in India. So they are basically emulating what US and Europe is doing. Now, in King Faisal Hospital, we are developing a local manufacturing program. We hired a scientist from UK who is actually working in manufacturing CAR T product, and we have successfully manufactured the first product, but we haven't infused this in human yet. We have ongoing 2 clinical trials, one for lymphoma, one for leukemia. And the process is not really sophisticated once you have this machine. And it's not very expensive, by the way. And the machine is very nice. And you basically buy the CRISPR gene construct, which is the most expensive part of the process. And if you have a gene editing facility, you can actually manufacture this locally. And you can do gene therapy for anything you want. You can do CD7 and treat T-cell lymphoma. That's still ongoing and experimental, of course. Just a few days ago, we infused the 100th case of CAR T in King Faisal Hospital, and of course, this is a celebration. There are more CAR T program opening in Saudi Arabia, in National Guard Hospital, our neighbor here in Riyadh. They actually treated their first patient in 2023. And I want to tell you something. Here, the CAR T program started in King Faisal in 2019. It took them approximately 2 years to infuse the first patient. That tells you just how much preparation it took for the hospital to prepare for infusing the first patient. I think that's all what I have, and thank you so much.
Thank you very much, Professor Ayman Saad, for this excellent presentation, and congratulations for your excellent work. Now, any question for Dr. Ayman?
Dr. Ayman, more than welcome in Ain Shams University. Next year, we want to see you physically, please. Ayman, you did a great job. My question related to home manufacturing in Saudi Arabia. What about the cost?
It is not terribly expensive. We get the product, we get the construct from a manufacturer who sells it for a few thousands, maybe 3,000 to 5,000 per construct dollars. So that's much less expensive than the CAR-T. In Egyptian dollars, it's going to be, we're talking about still maybe 100 to 150,000 Egyptian pounds. The manufacturing process itself is not terribly expensive, in particular if you do this on a larger scale, if you are manufacturing for 10 patients, because the whole cost comes from, first you buy the machine, it's not very expensive, but once you have the machine, the reagents and the tools themselves are not terribly expensive. Thank you for the compliment.
I have one question. Thank you very much for this very interesting presentation. So I'm also really impressed by your experience in establishing a center for CAR T-cell. So my question is, from your experience and knowing how the healthcare system in Egypt, it's going to be more challenging in terms of resources or for the CAR T process itself, or you believe it's going to be more challenging in terms of the required supportive care, the teams, and everything that's surrounding post-CAR T?
I believe both can be challenging, but can be doable. Technically, I believe Egypt should have the capability to do this. If you have a cell therapy lab that can do transplant, I don't see a reason why not to do the same thing for CAR T. It is not a highly sophisticated process. That's one. The clinical part can be challenging if there is no strict system in place, because these people get cytokine release in the middle of the night, and they can die by early morning if no prompt action was taken. So we hear, for example, here, we have a dedicated CAR T service, and it is required to call the consultant in service in the middle of the night to sign off in the tocilizumab order or steroid order. So it is not something we take casually. So this is something that it must be in place to make sure that this can be done safely.
Dr. Ayman, in the last 2 years, we have a lot of discussion about introducing CAR T-cell in Egypt. To what extent do you think it will harmonize with the established stem cell transplantation, which is already established in different leukemias and lymphoma?
I believe they should be complementary. There are patients, unfortunately, we cannot do stem cell transplant for people who have active disease or big residual disease. We still do autologous transplant in people in CR2 lymphoma. But we're always challenged by people who have refractory disease, because we know that neither auto or allo is going to help with these people. So I think it's a complementary. And I want to tell you that CAR T is not magic. Sometimes it fails and we have to do stem cell transplant afterwards. But if you look at statistics of the autologous transplant being done for lymphoma, it has dropped actually significantly over the past few years, because more people now are doing CAR T in people with first and second relapse. This hasn't happened for myeloma yet. I personally think it may happen because I think the outcome is going to be very close. So I believe they should be complementary, but that's an evolving question.
Thank you very much, Professor Ayman Saad. Thank you very much. And now we move to the next presentation, Navigating the Frontier of Myeloma Management, a spotlight on the recently approved drug therapy by Professor Mohammed Musa, Professor of Clinical Hematology and Head of the Clinical Hematology and Transplantation Unit in Shams University. Dr. Mohammed.
Thank you, Dr. Logna, for inviting me, Dr. Khaled, Dr. Shami. It's a pleasure to be with you all here in this international conference. My presentation will be about the multiple myeloma. I think multiple myeloma, we are now in a new era of multiple myeloma. Every day there is a new guidelines for that. Myeloma is a problem from last decades. the progression or the free survival is only 30%. With the progression and the progress of the treatment, it improved till 45%. Then the new era came. We started more than 70 years ago by the melphalan, which is still present till now, and the cornerstone of multiple myeloma treatment. Then steroid came, and the autotransplant, and the high-dose DEXA, and from 2015, the era of the monoclonal and the new target therapy started by the appearance of taratumab, ixazomab, leletazumab, and benovenostates, and also carfilzomab. And then the new era came again by the bispecific teclistamab in 2020. So all these drugs are available here in Egypt, and this is a good sign for us to find every drug you need for your patient. The goal, the multiple myeloma disease, there is a second most common hematopoietic malignancy all over the diseases, and you know that disease starts by MUGS, then with time, active myeloma, to give the patient first-line treatment, then the patient gain in remission, relapse, give treatment second line, relapse, give treatment till the patient is refractory and not responding and die. So the problem, how to extend the survival by prolonging the progression-free survival interval between the periods of the disease with minimal toxicity. How to select the appropriate induction therapy. First of all, you must consider the patient. He is not transplant candidate, he will take induction maintenance. If he is transplant candidate, it will induction, stem cell harvesting, stem cell transplantation, consolidation, and maintenance. The problem to select your patient from the start. NCCN guideline changes every day. This is version 1, 2024. We are still in 23. And this version was 1 month ago. And in this version, the recommended first-line treatment is bortezomib, lenalidomide, DEXA, and you can use all these combinations in the new era of myeloma. Also, if the patient is not candidate transplant, the first-line treatment will be bortezomib, lenalidomide, DEXA, or DARA, lenalidomide, DEXA as a category 1. Also can use DARA, bortezomib, melphalan, prednisolone as a category 1. So what to decide to take the patient will be a problem. Optimum therapy for transplant-eligible patient, you need to know your goals of induction therapy. First, high response rate with rapid response. You need the depth of the response with minimal residual disease. Improve the performance status and the quality of life of your patient. No limit peripheral cell mobilization for younger age. And how deep the response should be we aiming for. We need to put the patient in a very deep response to prolong the survival. This is the most important. So the induction regimen in transplant-eligible lesion, it was a problem. What to decide to take, VTD, VRD, VCD, and the problem was competition between all the drugs. First phase 3 trial was comparing our DEXA with or without bortezomib. And they found that by adding bortezomib, they improved the progression-free survival and the complete response rate by more than 80% 5-year survival rate by adding bortezomib for the patient. In the evolution trial, combination of VD with cyclophosphamide or lenalidomide was a problem. Which to decide? You take the patient VRD or VTD according to the patient. They found that VRD is better a little bit than VCD in the patient. Also, by adding Kyprolis, carfilzomib, by lenalidomide, DEXA, they demonstrate a high rate of MRD negativity response in all the studies which were done all over the years by all means. And KRD was effective treatment in newly diagnosed multiple myeloma patient, regardless of the patient is eligible transplant or not eligible transplant. And this was a very deep trial, and this trial, the high rate of 2 years at progression-free survival was very high. They reached more than 96% of the patient overall survival. Also, with maintenance by KRD post-transplant, they provide the patient with longer progression-free survival and higher MRD negativity rate than lenalidomide as the ATLAS study was done. So early monitoring of KRD-directed risk-adapted approach suggests KRD maintenance is potentially effective treatment for lenalidomide alone. So from this study, KRD demonstrated encouraging efficacy in newly diagnosed multiple myeloma, including a high rate MRD negativity, durable remission in phase 1 and 2. Also, KRD is effective treatment for newly diagnosed multiple myeloma patient regardless of transplant eligibility. And the safety profile of KRD in newly diagnosed multiple myeloma consisted with a known safety profile of carfilzomib. KRD is offer an effective backbone strategy for transplant eligible and ineligible patient and the patient in high risk cytogenetics. MRD directly risk adapted KRD could potentially be more effective than lenalidomide alone as a maintenance therapy for this type of our patient. Cassiopeia study, and this is the first time to use DARA, adding it to the VTD. As it's in European form, you know that the European group used thalidomide, but the American used lenalidomide. And this study was comparing adding DARA to the VTD as the standard of care, and by adding DARA, deeper response followed 2 years two cycles post-transplant. They found that there is significant more patient achieved MRD negativity with adding DARA to the VTD following induction, and after 2 cycles maintenance post-transplant, with 45% increase in the rate of MRD negativity by adding DARA. Also, they found by adding DARA there is high rate of progression-free survival, 53% reduction risk of disease progression or death, and after 1.5 years, they found that the progression-free survival by adding DARA is 93% for the patient. So, for the safety, there is nothing at all. DVTD has a manageable safety profile, doesn't decrease overall toxicity, and also similar treatment discontinuation rate by adding DARA, similar rate of grade 3 and 4 infection, no new safety signals was reported. So, what to do for the patient for multiple myeloma for maintenance post-transplant? This is a big problem. You need to get progression-free survival advantage, overall survival improvement, and the most important is the toxicity of the treatment. The immunosuppression, the lenalidomide, question mark with secondary primary malignancy, and the quality of life with the patient. Unclear whether all the patient will benefit from maintenance or no. Unclear which agent and duration of therapy. So, how to compare? The studies started by using lenalidomide comparing with the placebo after transplantation. And they found that by adding lenalidomide maintenance post-transplant, overall survival was increased and even improved the survival for the patient compared with the placebo. Also, CALGB 100-104 lenalidomide versus placebo maintenance, they found that the progression-free survival and time to the next treatment analysis found that there is improvement by adding lenalidomide as a maintenance for the patient, and overall survival also increased by adding lenalidomide as a maintenance. Okay, what will be the option for optimal therapy for transplant in ineligible patient? And we are knowing that the multiple myeloma disease is a disease of elderly. Most of our patients more than 70 years old. What we will give them? The myeloma in elderly is a big problem. Why? Disease comorbidities with high risk, frailty scores based on age, comorbidity, cognitive and physical condition can predict mortality and risk of toxicity. And what will give you the goal of the therapy? To give 3 drugs or 2 drugs. We know that the combination in the multiple myeloma is the best way to treat myeloma. So the best will be giving 3 drugs or even 4 drugs if the patient can tolerate. Duration of the therapy is important even in elderly. So suggested empiric treatment for the age-adjusted dose reduction for the multiple myeloma patient, they subdivided the patient according to the age. Group of patients less than 65 years old, and they will be candidate for transplant. If they are not candidate for comorbidities, they will be in this group. Another group will be from 65 to 75 years old, and the last group will be older than 70 years old. DEXA will be modified according to the age. If they are young age, you will give 40 milligram, and also the middle age, but the older age will give only 20 milligram. Melphalan dose will be in the younger and older will be only 25 milligram per kg, but in older group will be 0.18 milligram per kg. Cyclophosphamide, all of them you will give first two groups 300 milligram, but the older group will decrease to 50 milligram. Thalidomide 200, 150 milligram. And also lenalidomide according to the performance of the patient. The young patient will take 25 milligram for 21 days, but the older one will take from 15 to 25, and the old age will take from 10 to 25 according to can tolerability. Based on the combination in elderly, we can find that the best combination in this group of patients is MTP, or we can give them PVMB also, as we're getting good response for the long-time patient. First study, and this study was comparing lenalidomide DEXA versus melphalan with prednisolone and thalidomide, and comparing them to lenalidomide with low-dose Decta in this patient for unlimited time. And from this trial, they got that continuous RD improved the progression-free survival versus MPT or 18 cycle of RD for newly diagnosed multiple myeloma patients, regardless of age. Median and 3 years old progression-free survival extended with RD compared with the other groups. Also, 3 years overall survival extended with continuous RD compared with MPT, whether patient was younger or older than 75. And the analysis based on age consistent with the overall trial results. Toxicity profile for RD is similar among patients 70 years old or younger or older than 70 years old. But what to do for the duration of therapy for transplant in eligible patient? We give short duration, long duration, or kill disease progression. With this trial, they comparing the MPT compared with thalidomide maintenance with only MBR and R maintenance in transplant eligible patient. And they give in this trial melphalan, prednisone, lenalidomide as maintenance with lenalidomide, comparing it with melphalan, prednisone, thalidomide, and maintenance with thalidomide. From the start, you will find the most important problem with thalidomide is the peripheral neuropathy, which will happen in your patient. So from this study, they find that MBTT is similar in efficacy to MBRR in initial treatment of transplant eligible patient. The problem will be a lot of patients discontinue caring due to induction and maintenance, and discontinuation also was due to the primary peripheral neuropathy which happened with thalidomide in these patients. So, lenalidomide maintenance therapy generally well tolerated and manageable hematological toxicity in the old age group of patients, comparing it to the thalidomide. For the relapsed refractory multiple myeloma patient, they are very poor group of patients. They finished all line of treatment. And for that, we can find that the NCCN guidelines, the last version, which was 1 month ago, they decided category 1 as carfilzomib, lenalidomide, dexa, or DARA carfilzomib, dexa, or DARA lenalidomide, dexa, or exazotumab with carfilzomib, dexa. And all of them are category 1. Also, as the resistant bortezomib refractory group, we can use DARA, pomalidomide, dexa. And with the new era of DARA subcut, markedly improved for tolerance of the patient. As you give the patient an injection subcutaneously in 5 minutes or 15 minutes and go home with no at all problem of the injection and infusion time. And also for the previously treated patient with more than 3 lines of treatment and even relapsed refractory patient, we can use the protocol of carfilzomib, dexa, category 1, or elotuzumab, lenalidomide, dexa, category 1, or exazumab, lenalidomide, dexa, category 1. Also, there is a new combination of DARA with bendamustine, and also the use of venetoclax with dexa plus or minus DARA in the patient with translocation 1114. And the venetoclax in this era will make a marvelous era for the multiple myeloma patient. Boma with bortezomib, dexa for lenalidomide refractory multiple myeloma, and in this study they was comparing adding DARA for bortezomib, dexa, and they found that this combination is marvelously even with good response for this type of patient with good result. And by that they found that the PUMA adding to the bortezomib, dexa associated with high response rate 95% in the patient with relapsed refractory and resistant to refractory to lenalidomide by adding PUMA. And the PUMA in this area is very marvelous. The problem in Egypt is the cost of the PUMA. Weekly administration of bortezomib with dexa enhances the tolerability and the convenience of the treatment, and PUMA bortezomib dexa associated with manageable adverse event and primary mild cytopenia happen to the patient. So it's a good combination, but the problem in Egypt will be the cost. Also, in serious study by DARA as a monotherapy in heavily pretreated multiple myeloma patient, and in this study they comparing adding only DARA maintenance and starting from the start with maintenance till disease progression. They found that the DARA in combination with lenalidomide, dexa with overall response rate 100% in part 1 of the study, but look, the complete response rate is 31% only. But overall response rate is 100%. And in part 2, it's 86%. And this means that by adding this combination, you will find 100% cure rate for the patient. Also, a new era for the use of BTK in the era of multiple myeloma, the use of protein alone or in combination with DEXA in relapsed refractory multiple myeloma. The protein will be added in everything. It was in CLL, now in multiple myeloma, and in lymphoma. We now, and within a while, mantle cell lymphoma, and the group of BTKs all will be this era. And in this combination, there was starting an open label phase 2 dose escalation dose for the protein, and they found that the use of 560 milligram in coherent 2, this is the best combination comparing it, and by the coherent 3 and 4 with the high dose 840 milligram. A protein with DEXA with overall response rate more than 60% of the patient. For this type of the patient by the high doses, but the high dose was 840 milligram. It's very big dose. And in this, they found that the more progression and more progression-free survival and improvement in the survival, oral treatment at home, refractory, relapsed, resistant multiple myeloma patient. And from this study, they found that a protein with weekly dose DEXA appears safe, promising activity in heavily treated patients, complete response rate 25%, sustained stable disease at 25%, and progression-free survival was 6 months. Safety profile with adverse event rate consistent with this seen in other histology and other drugs, as the protein is very safe and less side effect. Enrollment of additional people in this studies, in this study, that they can improve the survival of the patient. And further study of progressive combination with backbone agent as a warranty انها تشتغل وتبقى most important for the time being. The additional even currently in early phases of development, the use of protein-bifid and flizonumab, and also ruxolitinib and lexalostat, selinexor, as Dr. Azad said, and also venetoclax. Venetoclax will be a more powerful drug and will be a good in multiple myeloma treatment. And thank you.
شكرا دكتور محمد على المحاضرة الشيقة ديت. Very clear actually. Multiple myeloma is very challenging now. Any question?
Dr. Mohammed, honored to be with us, with Professor Dr. Assam Abdul Wahid. Always, we are one team. My question related to multiple myeloma is a heterogeneous group, okay, high risk, low risk by cytogenetic. Now we notice that the age or the median age of our population is younger, at least 10 years.
Yes.
Yeah. As an expert, your opinion to introduce DARA. We have an experience with DARA in Ain Shams University. To introduce DARA in transplant eligible, in high risk, the best position. This is the first. Second question, your experience with Kyprolis, with cardiac toxicity, especially AF.
Yeah, for the DARA, I think for high risk group and young age group, it would be a best to give from the start, first time treatment. The problem is the cost. But I think with the use of the subcut now and the decrease of the cost by more than 75%, we can add it as a first time treatment for high risk and young age group. And also the problem with DARA was previously the yield of the collection for the harvesting for the stem cell transplantation. But with experience, there is no problem at all by adding DARA the first time. This is a problem. The other question about the carfilzomib toxicity, for me, I didn't see any patient with cardiac toxicity, even with the Egyptian population. I use Kyprolis more than 5 years now. I didn't find any patient with cardiac toxicity. If any one of the panel find the patient with cardiac toxicity with Kyprolis, he can tell me. Egyptian population is a very different population from others. In CML, we have the youngest age. I transplanted 3 months ago, multiple myeloma patient, 24 years old. 24 years old. Yeah, it started by osteolytic lesion, vertebral osteolytic lesion only. He take radiotherapy and low-dose treatment, and then he relapsed with very aggressive form, 24 years old.
Can I have one question? Thank you very much. Very comprehensive presentation indeed. I enjoyed very much, especially that myeloma is not my top area of expertise. So I have a really question that always a situation when you find a patient coming with considerable amyloidosis, what would be your preference?
In Egypt now, we're dealing with amyloidosis by first line DARA. We are giving DARA BRT in Egypt now, yeah. And the awareness of amyloidosis now improved. Janssen make marvelous awareness for the amyloidosis between the cardiology and nephrologist, and
Very
this
good.
is improved the diagnosis. The problem was privacy in the diagnosis, but now by the awareness, markedly improved.
Fantastic, thank you.
Yeah, thank you.
دكتور محمد بدر. الكرونيك ليفر ديزيز في مصر يعني فيري كومن. فانا لو عندي عيان ترانسبلانت اليجابل وفي هايبرزبليزم فدايما في البيز لاين بتاعه بانسايتوبينيا. ايه راي حضرتك الاندكشن هيكون ايه الموست سيوتبل؟
You want the induction from multiple myeloma from the start?
هو مالتيبل مايلوما والبون مارو بايوبسي طلع هايبرزبليزم وطبعا فلتريشن بالمالتيبل مايلوما بس كليفر كومبنسيتد.
I think VRD will be best for him. Yeah, VRD will be, there is no side effect for the hepatic toxicity. The problem with VCB, the problem with VCB is that Doxan is very injurious for the liver, but R is no problem at all. Yeah, but the problem will be during the transplantation, the performance status of the liver during the transplantation. If his child B or C, he will not be transplanted. If child B and the performance status of the patient is good and can tolerate, we can proceed, but will be with low dose methanol.
شكرا. Okay, thank you, Dr. Muhammad, for this elegant presentation, and I think now we came to the end of this session. Thank you.